N-nucleotide addition can also happen when we have blend ends where, trying to remember what N goes with what. So AT was here. Okay, so these are our N's, and N-nucleotide addition has come in here to add these bases in red that weren't scripted before. Just plug those guys in. So TDT is the enzyme that does this. This does non-scripted N-nucleotide addition. So there could be P-nucleotide addition that is scripted by the overhangs. They find where A's, T's, G's, and C's overlap, and we put them together. And nucleotide addition happens when TDT randomly adds. So this thing right here, NHEJ, this stands for non-homologous end joining. And this is a DNA repair mechanism that can happen during any type of DNA repair. But it can also happen during crossing over events, making gametes, meiosis, or spermatogenesis, where pieces of chromosomal information that have overlapping pieces of information, right, nucleotides that are similar to each other can swap and stick to each other. So sometimes some of the enzymes that are involved in this type of repair are involved in this process too. They can be used here as well. This slide is pointing out to us that sometimes information is encoded in different orientations inside our genes. And the point of this, the take-home message of this, is that even if information isn't always encoded in the same direction, when RAG1 and 2 and all of the accessory enzymes come together to make joints, they can put the information in the right orientation. Right, so in this case, we're looking at some variable regions, and with encoded in this variable gene segment, you have information that's encoded left to right in variable segment 1 and in variable segment 3. But if we look at variable segment 2, it's the reverse. It's actually read right to left. And the signal sequence joint, the heptamer nonamer, is in the opposite direction too, right? It's on the opposite side of the information. If the cell actually wants to use this region, it can, but it doesn't have to. By making hairpins, by coiling the DNA sequence around each other, it can change by physically changing, by physically moving the DNA, swapping the direction that it's going in. You can make the cut at the appropriate site and rejoin in the right encoding information, or in the right encoding direction if you want to. It's okay if it's not. So this is talking about orientation, signal joints, things in the right and wrong direction. We're going to physically move the information around instead of cutting and pasting and then some machine turning it around. We're just going to use the coiling of DNA and the wrapping to create a hairpin to get everything in the right direction. Okay. So now you have seen that there are multiple segments of information that can be mixed and matched for variability. You've seen that sometimes there's nucleotide addition that can happen because the ends that are cut have overhangs, and those overhangs allow us to seal things together, and then ligase finishes that off. That would be scripted. That's P-nucleotide addition, happens to join the ends together. There can also be non-scripted nucleotides added to join the regions together. So this slide is to give us an idea of what are all of the events, what are all of the things that are taking place that allow us to make billions of different combinations. So the first one, right? Each region, with the exception of the constant regions, they aren't involved in binding antigens, so it doesn't matter. The regions that are involved in flexibility and antigen binding, there are multiple different gene segments that we can use. P-nucleotide addition will add the joints together. It will find little overlap pieces. Oh, hey, this guy's got a C and this guy's got a G, so we're going to C and G, and then we'll fill in whatever is left over. So that's P-nucleotide addition. That creates some randomness in how the joints are made. Sometimes, I forgot to mention this when we went back, and I think there actually is a picture of it. There can also be trimming of the overhangs. So let's say cell doesn't like, there's a T and a C and a whatever, it's like, oh, these don't mismatch. There's an enzyme that can come in and chew off nucleotides as well to help make the joints. So you can have scripted joint making, P-nucleotide addition. You can have exonuclease trimming, cutting off overhangs or removing segments that you don't want. You can have N-nucleotide addition, so non-scripted, just randomly put some A's, T's, G's, and C's to plug it all back together. So that adds to the randomness. And then the last thing, right, the last one, there are 5 mechanisms, continues up here. It's called combinatorial diversity. And what that means is each heavy chain is made randomly, each light chain is made randomly, and what heavy chain gets paired with what light chain is also done randomly. Right? So you make the heavy chains, you make the light chains, you make them independently of each other. Then you take those and you put them together structurally. Right? So you pick a random one from the pile, you pick a random one from the pile, you stick them together, you get a combination. And you pick another random one, right? So I'm going to take blue heavy chain and mix with yellow light chain. Then I'm going to take red heavy chain and mix with purple light chain. Right? Mixing and matching lots of different versions. Yes?
Are all 5 mechanisms random, or is there some
P-nucleotide is the least random because it's guided by overhangs, but everything else is random.
How does the P-nucleotide addition still add to variability and randomness?
So you may be, okay. Alright, let's say I have a segment. You got one side that's, you've got another side that is nope, I don't want to start that way. I want to start okay, so here's one end. Here's the other end. I gotta sew these two pieces together. C and T don't match, right? So exonuclease trimming might cut them away. Okay, so if I take these out, can I match A and A? No, right? So I gotta cut these out. Can I match this? Yes, I can. So there's going to be tweaking that happens with the sequences, and you don't necessarily have to cut these out. So a weird thing that could happen is the cell could decide, right, I can't remember what was here now, but Doesn't matter. If I took this, right, I got T, A, G, C. What can I make match? Where do I have the most matches? We could overlap in places where there aren't really overlaps, and then there would be intervening sequences, and those would get chopped out. So the way you realign the overhang doesn't have to be perfect. So that's why it can add variability. Someday I'll draw myself a picture of that. That makes more sense and put it in here. Okay. So numbers. Numbers of V regions, numbers of D regions, numbers of J regions, possible combinations that we can make for heavy chains. Possible numbers of kappa, light chains that I can make. Possible numbers of lambda, light chains that I can make. Then I take a heavy chain and I randomly could pair with a kappa light chain or a lambda light chain. Billions of combinations without thinking about P nucleotide addition, exonuclease trimming, and N nucleotide addition. So this is just raw numbers. How much variation can I make? Yes?
So is it kind of safe to say P nucleotide addition is somewhat of a modification of something that was already there, and then
Yes.
N nucleotide is more
Random, completely random. Yep. Yeah, we're going to work with sequences that are already there. But we could align things in ways that aren't perfect for P nucleotide addition. Okay, so this is just, right, raw numbers. But then there's also the randomness of how will P nucleotide addition put everything together. Sometimes it will be exactly the way it's scripted. Sometimes it won't be. And nucleotide addition can happen between any of the segments. Exonuclease trimming can happen within any of the segments. We can't predict how those ends will be joined together in those ways. So that adds some more variability to the process. Right, but the 5 ways make sense. So multiple segments, multiple V's, multiple J's, multiples V's, D's, and J's. Sometimes there's scripted addition, sometimes the script gets tweaked a little. Sometimes we chew up the ends, sometimes we add ends. Right? So these are more random, these are a little less random, but could still be weird. Right? And then combining. Random heavy chains, random light chains. And I can mix and match. I can have a heavy with kappa, I can have a heavy with lambda. Right? So I'm making all these different versions and they're being made at any given time inside our cells. Okay, so maybe you've thought about this, maybe you haven't, but this is happening inside somatic cells, right? So we have our full chromosomal complement. You have a chromosome 22 from mom. You have a chromosome 22 from dad. And you have to pick, your cells have to pick which chromosome am I going to try to rearrange first. So if your cells decide to choose dad heavy chain to rearrange first, mom heavy chain information will be temporarily silenced while that rearrangement is going on. So this is a process that we call allelic exclusion. It means that inside the B cell or the T cell, right, you have 2 copies of the chromosomal information that you're going to use to make your heavy chain or your light chains. The cell has to pick one. It cannot be doing at the same time. It cannot say, oh, I got 2 copies of chromosome 22, and I'm going to try to rearrange both copies and see what I get out of it. The cell has to choose. It's forced to choose. So we're going to pick dad's first. Okay, great. We're going to get our D's and our J's, and we're going to clip our RV on there. We're gonna rearrange that guy. We're gonna make a transcript. We're gonna try to translate that transcript, and we're gonna see if it makes a protein. Mom's information is silenced. It cannot be accessed. RAG1 and 2 cannot be going there, it cannot be cutting, it cannot be trying to rearrange. It's off. We're going to test the heavy chain that we made from Dad first and see, does it work? If it doesn't work, we can try to rearrange Mom's information to make a viable heavy chain. Right? So we're not trying to rearrange all of the chromosomal information at once. The cell does, it's a very ordered, stepwise process. We try to rearrange one chromosome first, try to make a viable heavy chain. If that does not work, then we go to the backup chromosome. And we use that information and we try to rearrange. We make a transcript, we try to translate, and we see if that works. If that doesn't work, if rearrangement of heavy chain from either of the pair of chromosomes is no longer successful, the cell is forced to undergo apoptosis. Okay? And then we gotta so we build a heavy chain first. Then we gotta build a light chain. So let's say rearrangement for Dad's heavy chain works. You get a viable heavy chain, viable protein inserted in membrane, sweet. This activates RAG1 and 2 again to go to kappa or lambda information and start rearranging. Now, if we choose kappa to rearrange, lambda is silent. So we try to rearrange kappa on Dad chromosome. Oh, that failed. Okay, we try to rearrange kappa on Mom's chromosome for Mom. Try to rearrange that. Oh, that failed. Now we can go to lambda. We can go to lambda and try to rearrange Mom's chromosomal information this time. Maybe that works. Okay, it works. Okay, so we're going to make a transcript. We got our protein. Now we got Mom light chain, Dad heavy chain. Stick it together to make a new protein. So it's a very ordered process. We try to build the heavy chain first. We have 2 chromosomes, right, to pick from for that information. We're going to rearrange one at a time, though. If one is successful, the other one is silenced. While we have a heavy chain and we're working on light chain rearrangement, there's a placeholder that binds onto the heavy chain. So here's a heavy chain that came from one of the chromosome pair that was successfully rearranged, successfully transcribed, successfully translated. I got my beautiful little BCR heavy chain hanging out in a membrane right now. This is not a complete molecular machine yet. It still has bits and pieces anchoring into the membrane, but it has not been fully hooked up to Ig alpha, Ig beta, ITAM holders. And this is not ready to bind antigen yet. And so these special placeholder protector proteins called lambda 5 and B get added onto the heavy chain to act as a protector. And the reason for this is that the heavy chain is part of an antigen sensing complex. And we don't want this to bind onto something and to trigger signal transduction or changes inside the cell before the molecule is ready. So this acts as a protector. It keeps this thing from binding onto things and turning on signal transduction and getting the cell to try to do a behavior it's not ready yet. It's still maturing. So this placeholder takes care of the heavy chain molecule while we wait to receive a functional light chain molecule. So let's just make sure that the heavy chain stays there. It's good. It's waiting for its new light chain partner. So light chain rearrangement starts to take place. So what's being shown in this diagram are what happens in the outcome. How does all of this work out in a timeline? Okay, so we're going to start back here. We have different names, different developmental stages that we give our B cells and our T cells based on the stage of construction they're in building their BCR or their TCR. So So you've got your lymphoid progenitor that is going to activate specific gene pathways that are going to help dictate, do I become a T cell, do I become a B cell? There has to be a decision made early on that decides which I'm going to be. In this case, we've decided to be a B cell. Now it's time for us to start building a heavy chain. So RAG1 and 2 become activated. This first stage where we start to build a heavy chain, we're truly kind of decided we're going to be a B cell because we're going to make a BCR. We call this thing a progenitor B cell, sometimes also called a pro B cell. This guy's got RAG1 and 2 activated, cruising along, looking for heavy chain diversity and joining regions to clip together. If we clip this together functionally, now we have a chromosome that has the DJ pair. We've got to figure out what V we're going to add. A neat thing happens during this process. This cell, once it has successfully done this, is going to rearrange a V onto that DJ. If you get a successful VDJ rearrangement, the cell, there could be two outcomes that happen for my VDJ. One is I have successful VDJ rearrangement. Right? So everything worked out great, smooth, simple, easy. My two DJs came together, that was great. We added a V on, it was good. We can build a heavy chain, yay! It's productive rearrangement. So now what happens is I have my heavy chain and the surrogate light chain is paired on there. Right? So that shows us what happens if everything just works perfectly when we're trying to build our heavy chain. The alternate right here shows us what happens if that VDJ rearrangement doesn't work out the way that we planned. So I've got a VDJ that rearranges. We test out that protein, but it's non-productive. Right? So we've done VDJ rearrangement. We transcribe, we try to translate, but what we got didn't work out. Right? So what are we going to do? We have another chromosome that we can VDJ rearrange, right? So that's what we're going to do. We don't want to just give up. I don't want to give up. I'm going to try to rearrange the other chromosome information to get a successful VDJ. In this case, so let's say we chose dad chromosome and it did not work out. We're going to go to mom chromosome. Try it out. VDJ rearrangement. Sweet. It worked. Because we have a heavy chain transcript with the light chain placeholder. In this case, we rearranged. It was non-productive. Then we went to mom's. We tried to rearrange. It was non-productive again. So if we try to rearrange mom, heavy chain, and it doesn't work, we can go to dad's heavy chain. But if dad's heavy chain doesn't work, we're out of heavy chains, we're out of chromosomal information to rearrange. We will be forced to undergo apoptosis. Right? So always the end game. If you're out of information to rearrange, you're going to try one chromosome first. If it doesn't work, you go to the other chromosome in the pair. If that works, great. If it doesn't work, you're forced to undergo apoptosis. Yes?
Do most cells even get to the pre-BCR stage?
No. Usually everything fails in here. But if we successfully put up a heavy chain, now we got to do light chain rearrangement, right? So some cells are going to choose kappa first, some cells are going to choose lambda first. Same process has to happen. We're going to try to rearrange dad kappa chromosome information. Did it work? Yes or no? If it doesn't work, then we try mom's. Did it work? Yes or no? Oh, now we can't use kappa. Let's try lambda, right? So we keep going through the series of possibilities until we get a viable outcome. If you never get a viable outcome, the end game is always death. Yes?
So how are these
So in the beginning, it's can you make a transcript? Can you make a viable protein that can be translated and inserted in a membrane and bind to the B pre B lambda 5 placeholder? So the placeholder acts as the test in this case. First, do I get a transcript? It's in a membrane. Okay, great. Can B pre B and lambda 5 bind onto it? If they can, that's good. Because that means if we build a functional light chain, it will be able to bind on. If these guys can't bind onto here, the cell dies. Because it doesn't get the input, it doesn't get the cell signaling that tell it it's okay for you to rearrange a light chain. So it's all about signal transduction, and touching, binding this receptor and what state the receptor is in tells the cell, is it okay for me to proceed, or is it not okay for me to proceed? So that's like the checks and balances that happen during the building. Does this make sense? I know it's weird to think about, right? So you've got duplicate sets of information. You got a set from mom, you got a set from dad, and you can use that information, which is pretty cool because it means we express chimeric information that came from mom and dad to build our VCRs and TCRs. It's kind of neat. The same thing is true of our MHC receptors, and I'll show you that in the coming week.